And then we did another analysis, which is called a birth cohort, where we then, instead of just looking overall in the population at a single point in time, we take children who were born in different years. So we're sort of controlling for age factors and look to see how the trends differed for children born in each successive birth year. So on this graph, each of the lines represents one birth cohort as that birth cohort ages from 1 to 9 years of age. And what's striking about this birth cohort analysis is that there's no crisscrossing of the lines. Each year is above the line before it, representing that time trend that each birth cohort is at a higher risk at every age. Comparing the 1990 birth cohort to the 2001 birth cohort and following up to age 4 or up to just before the 5th birthday, we see that it rose from 6.2 to 42.5. That's a 7-fold increase in a 12-year period. So I'm going to talk about this as a 600% increase over the background of 100%. And talk about what might be the causes. Well, first of all, what this means is a rise in the cases who are receiving services in California. And I wouldn't pretend that this is an absolute complete ascertainment, because we know that some children aren't in the system. They may have families who have the resources who don't go to the state for services. They use their own private wealth. There are people at the bottom of the socioeconomic ladder who tend not to avail themselves of services, and other groups that may be underrepresented in this database. Nevertheless, it's believed to account for about 75% of cases. So what is this increase due to? A number of different possibilities. The idea that there's more hope, that people are more likely to want to bring their child in if they don't think that this diagnosis is a sentence to lifelong institutionalization. I think that attitude has changed, because we know there are some therapies that do help with behaviors, and maybe not every child, but certainly the feeling about the potential is there. And then, of course, possibly it is a true increase in incidence. So we looked at data that we had from a variety of different sources, and we showed that the change in Diagnostic Statistical Manual criteria could account for about 120%. The trend towards younger age at diagnosis could account for about 24%, and the broadening in terms of practice to include milder cases about 56%. And then older age of mothers, because it turns out that older mothers actually do have a higher risk of delivering a child who will develop actually many conditions, but autism being one. These add up to 204%. So it's about a third of that 600% that we're trying to explain. The possibility of a true increase from these data then remains. And after we published, another group out of Columbia looked at a phenomenon we call diagnostic substitution, where back in 1990, maybe we would have called that something else. Maybe at that point, mental retardation. Now we would call it autism, because people are more attuned. So they also found about a third, a quarter to a third of the rise might be explained by that. And there might be some overlap of what they were looking at and what we were looking at. So we can't explain it all at this point. And this is a little thought experiment, which is that we see this rise and we see other things that have been rising in the last few decades, to maybe try and look at the whole of what's going on in terms of health and ADHD, asthma, obesity, diabetes, and other conditions. Is there some common set of factors, some environmental factors, that could play a role in all of these conditions? Now, I also want to talk about genes and environment. I'll be focusing mostly on environment, but it's important to realize that autism is not going to be found to have a single cause. And I don't mean even across the population, even within individuals. There are probably many factors that come together and eventually lead to the behavioral disorder we call autism or autism spectrum disorder. The little circles here represent what we call in epidemiology a sufficient cause pie. So the pie on the right with E, B, C, and F has 4 causal factors. And if you took away one of those factors, it would be an insufficient set, and you would not see the full-blown disorder. So genes, we know at this point that there is high heritability. A number of genes, a large number of very rare polymorphisms have been discovered that may account for less than, each one, less than 1% of cases, and collectively perhaps somewhere around 10, 15, 20%. We also know that there are some very common genes that are associated with higher risk for autism, too common to be purely the cause. So again, we have to think about genes and environment together. The genetic studies on monozygotic twins who have exactly the same DNA from both parents suggest about 60 to 90% concordance. If one has it, then the other would have it. People have mistakenly assumed that the environment, therefore, can only have somewhere from 10 to 40%, because they assume that there's not an interaction between genes and environment. But in fact, the environment could account for a lot more. So where are we in 2010? Genes, yes. Environmental chemicals, I'll talk about that a little bit in the next section of my talk. Nutrition, same thing. Microbiological agents, these are all valid hypotheses to be investigated. We know a little bit about the gestational and perinatal environment, and this is from those studies I mentioned earlier, the administrative databases. Because those data already exist, this is probably the one area that's been investigated the most. You don't need to get a huge amount of money to take a database and just analyze it. You don't have to go out and collect all the data yourself. And so then there are hypotheses about the fertility treatments, increasing use of assisted reproductive technology, maternal reproductive characteristics, medications that have changed our prescription and our consumption practices over time, and modern technology. All of these have been posed as things to investigate. So we started the CHARGE study, which stands for Childhood Autism Risks from Genetics and the Environment. Back in, we designed the study in 2001, and in 2003 we went into the field to find out what are the environmental causes and how do they interact with genes, and also are there certain subsets, certain types of autism that might follow a different kind of mechanistic pathway, and maybe different causes might go with different types of autism. Looking at this biologically, not just behaviorally, but behaviorally also. So looking at those regressive versus early onset cases, looking at kids who have immunologic dysfunction from those who don't, and so forth. Well, where do you begin? And people throw up their hands and they say, the environment, that's everything. Where are you going to start? So looking at both what we knew about autism, but also what we know about other neurodevelopmental conditions and what we know about environmental neurotoxins. That was how we sat down and came up with our first list, which included pesticides, metals, organic pollutants, medical procedures, and nutritional factors. And all of these arrows point to the ways that we can measure those things in a study. So we can collect biospecimens, and we can use blood samples to measure levels of these pollutants in the body. Turns out that you can take hair, and many mothers save the baby lock, the first haircut, and you can measure metals. We can look at interview data, we can look at medical records, and in California we actually have an amazing array of existing databases that pertain to exposures, including traffic exposure, air pollution, pesticides, and so forth. So we thought we would bring all of this together into one big study, and that's what we did. And we have, this is called a case control study, which means that we have cases who have the condition, and then we have comparison groups that we call controls. And so that's a key feature of this study. We used population-based recruitment. We didn't rely on a single or a couple of clinics that have certain specializations. We confirmed all of the diagnoses in the clinic with our excellent assessment team at the Mind Institute. And then we're linked to all these laboratories which the specimens go back and forth between. So the two case groups here are children with autism and children with developmental delay, but not autism or not an autism spectrum disorder. And then children that we drew from birth certificate files through a random sampling process where we also matched on age and gender and the general geographic area. So the geographic area is this area of Northern California, sort of surrounding the Mind Institute in Sacramento County, covering the green, the yellow, a little bit of the gray, and the pink there. And the eligibility criteria is 24 to 60 months, born in California, a parent who speaks either English or Spanish, and living within our catchment area. We actually had a little bit of Los Angeles involved in the early phase of the study as well. We do the ADOS, we do the ADIR, we do a medical exam, and we assess really a broad array of behavioral factors and medical factors as well. And then we have a very lengthy interview getting at medical conditions, residential history, lifestyle, use of household products, and so forth. And we're aiming at the prenatal period and the early and postnatal period. Actually, we begin 3 months before conception. And then, of course, specimens, which we collect not only from the child, but other family members. And then we get permission to get the medical charts and try and bring those in-house as well. So our slogan for the participants is, it's time for you to be in charge. And I'll start with the results on metals. So the goal here was to determine whether mercury levels in children with autism differed from the levels in children who did not have autism. These graphs, these histograms, show the distribution of the levels of mercury along the x-axis. It gets larger. It's on a log scale. That's why you have negative values in there. But what you see is that the distributions overlap a lot. And interestingly, you can sort of see the peak for the typicals is a little bit higher than the peak for the autism group and the developmental delay group. So we looked at that a little more closely. Some people were predicting it would be higher. Some said, no, it'll be lower. And we found that there was a reason why it was lower in the ASD group, and that is that they were less likely to eat fish. And fish is, in fact, the main source of mercury in the human body. Now, I do want to put a little disclaimer here, which is that fish is very beneficial for you. And it's also very beneficial for pregnant women to eat fish. It actually improves cognitive development in children. This has been shown in study after study after study. So the studies that looked at mercury in the beginning came out really mixed. They couldn't quite figure it out. Some studies showed a deficit for cognitive development and some didn't. And it turned out that if you control for the fish, you see that the fish improves the child's cognitive development, but the mercury actually causes a decrease in cognitive development. So what you need to do is eat the fish that are not high in mercury, and there are many, many websites that have information on what kinds of fish are the safest in terms of mercury levels. So that's cognitive development. Now what we're looking at here is mercury in the children, not in the mother at the time of gestation, and we see that the children were eating less fish, which is why they had lower levels of mercury. We also looked at some of these other sources of mercury, and these didn't differ very much. When we did a statistical model in which we related all of these things to their mercury level, we found that having an autism diagnosis showed really no association whatsoever, and that's what this little p-value over here of 0.75, this is not a statistical difference comparing ASD to typically developing children. All of the other factors here, except for the use of these personal care products that contain a little bit of mercury as a preservative in them. All of the other ones are significantly related, and I draw attention not only to the fish, but mercury amalgams in the mouth of the child if the child chews gum or has a teeth grinding habit. So just having the amalgams by itself didn't do it, but if they were constantly biting and chewing and grinding, they had higher mercury levels.